thanks to the organizers for inviting me to such a special place as Singapore. Unfortunately, I cannot spend much time here, or otherwise I will be divorced. And so next time I hope to see something around. While preparing my talk, I was thinking about what kind of things I could talk about, and I discovered that too many. And so what I'm going to try is to address some of the items that, in a way, constitute a new way of approaching complexity from a number of areas that try to build alternatives to the reality we observe, and in particular to approach the problem of what can be constructed, what can be evolved, what can be designed. And are there constraints that evolution has put into our capacity of changing nature, or there are instead, as it seems the case, a lot of barriers that we can actually break. What about this picture? You're wondering. Well, one of the themes, if you want, that connect really many of the researchers in complex systems is trying to find out whether or not there are general laws. Are there general laws of complexity? And we can rephrase that in the following terms. Why there are no monsters? As you know well, when the explorers reached the ends of the world in the old days of exploration, they went back with stories about the monsters that inhabit those seas, et cetera, et cetera. Those, of course, were the times of the maps filled with empty blank spaces, the terra incognita. And we know that when the exploration finished, the monsters disappeared, as far as we know. Just to be sure, this is an example. I'm referring to the monster of this site. Why we don't observe that? That's one question that you can formulate in terms of what is the actually, what is the possible, and why nature, why the biosphere, why human consciousness, if you want, is the way it is. So what about the big questions? Because one thing I learned at the Santa Fe Institute, almost the first day I went there, was the first thing you have to know is, what is your question? Don't come here with something like, I would like to make models of, no, what is your question? So there are very, very big questions that are not answered. Unfortunately, I'm not going to answer them today. Like how the universe originated, or why there's something instead of nothing. Although, you know, this can be rephrased, and you take into account that everything seems to indicate that the overall energy of the universe is zero. In reality, there's nothing, except that nothingness has adopted this peculiar configuration that includes us. What defines life and how life originated? Can we build artificial cells? How complex cells emerge? How did multicellularity arise? Is it possible to break evolutionary rules? That's something that 10 years ago, many things that we thought about aging and death, everyone will have been agreeing about this is not possible. And we have to rechange a lot of things. If we make a jump, and we go to that at some point in the talk, is the question of how far machines can go. What can we build in order to get something that is intelligent or conscious? And I'm going to try to convince you that although these questions are legitimate and very, very interesting, there are other questions that are already emerging in our way to that direction. Is technology the sixth kingdom? That's one question that I'm not going to address because we don't have time. We don't have time for anything, actually. But it's been conjectured that technology itself is kind of a new form of evolution, of evolutionary organism, if you want, that lives and thrives thanks to us. But it's kind of an entity that you can actually predict some kind of traits. Will we survive? I added that after Martin Ruiz mentioned this stuff about our final hour in the most dramatic sense, and you'll see why I'm addressing that. Okay, what's the umbrella I'm going to use? Several years ago, Eor Shazmati and John Maynard Smith, two very important and influential evolutionary biologists. I must say that, of course, because Eor is here, so I mean, I have to remember not making jokes about that. They wrote a book who had an enormous influence. You see there the citations over the years, and they never faded out. where they try to put together a very important material. We know that evolution is a fact, that it happened. It's either that or the devil built a whole thing to confuse us. Suppose it's the first alternative, and they were trying to put together is what are the relevant innovations, what happened in different moments in the history of life in our planet that represent a qualitative change. They made a list that is basically valid today. They were conscious that there are other things that could have been added or rephrased, but I think that everyone will be confident in saying that things like sex, life, of course, cells, or multicellularity and language are fundamental changes in the history of life in our planet. Now the thing is how that happened, and that's one of the big questions that we still have to understand. Other people have used different lists. So some people, like in this book by Nick Lane, add movement, sight, hot blood, consciousness as big transitions, and death. That sounds like strange, but death is a very, very important invention of nature. Without death, will we not be here? Interesting. In the other list here, you see, again, common items, but you put in this list, Rachel Novak puts the eye, photosynthesis, or parasitism. Parasitism is clearly a qualitative change in our biosphere and something that seems to be very, very universal. Goes from protocells to societies. So I want to use this picture, this idea that large, big transitions have happened in the biosphere, and ask myself, and ask to you too, to what extent can we revisit that, and to what extent can we reproduce those transitions, to what extent can artificially approach them, and is there anything different that can be done? So this is a slide I show to my students. I have this 4-year degree in bioengineering where against all the standards, we teach them a lot of evolution, a lot of molecular and cell biology, a lot of complexity theory. And I put here some of the big transitions, the major transitions, along with things that are going on now that have to do with new technologies that are being developed and new discoveries that are being made. And I want to go through some of them, not all of them, because we don't have time again. But as you will see, by trying to understand those transitions, we find out really, really interesting ideas and completely new questions. So first, what is life, and can we imitate it? Let me make a little historical parenthesis. The problem of what is life, it's an old one. And in the 18th and 19th century, there was a very high excitement about the potential of building lifelike systems, and those were the mechanical automata. This is an automaton that is still working. It's not like your iPhone that is in 2 years you have to change it. It's still working. It was built more than 150 years ago. It's in Neuchâtel, and it was built by a mechanical genius, Jacques Droz, who was an amazing engineer, and that built a number of really amazing automata. The idea that the machine could be lifelike created a huge amount of philosophical thinking and a lot of funny stories. Let me tell you one, because I love it. Jacques Droz, at the time, there was no rock bands, so there were automata makers who go on tour in Europe. And so he went on tour, and he made the mistake of going to Spain. Why I'm saying that? Because they went to Madrid, and there were, exhibition, but, the Inquisition was still quite active. So they find out that someone who was trying to build a human was heretic. So they put them in jail, both of them. So imagine the creator of the automaton and the automaton in the same cell for 2 days. So it was like giving the life that they were trying to deny to the automaton. And the story is full of that kind of things. So I'm going to talk about synthetic biology, and every time a journalist comes to our lab to ask about things, Frankenstein comes out. So we say in Spanish, that in Spain, that if you cannot win them, just join them. So Frankenstein, of course, it's a great metaphor. Now I'm realizing at this moment that I made a mistake of putting that particular video, because we are not going to pay any attention to what I'm saying, but try to concentrate. Frankenstein is interesting because it's not just the amazing, it's impossible that you are paying any attention to me. So, I'll try to concentrate myself. So in that particular book, it's not only an amazing creativity act by Mary Shelley, which was 18 years old, but the fact that the science at the time was completely percolating and permeated by the advances of science, magnetism, electricity, and at the time that Shelley was writing that, they had already been seeing all these demonstrations where one of the nephews, I think, of Galvani was making these experiments with corpses from people that had been hanged, putting electrodes into heads and corpses, and they could see that they opened the eyes or kind of tried to move or breathe. So from that to thinking that electricity has an energy that can communicate life, it's not a big distance. So hoping that you have been listening to me, there's one thing in the book that is quite interesting about multidisciplinarity. At some point, Victor Frankenstein makes this statement: I have not neglected the other branches of science. If you wish to become really a man of science, dots, I should advise you to apply to every branch of natural philosophy, including mathematics. The dots are very insulting comment about experimentalists, so I just tried to remove it. So synthetic biology means to work at the level of biological molecules. We just heard a very interesting talk where you can see the difficulties of that. That it's not trivial at all. I know that physicists tend to say, wow, this is very simple. But no, we are not always right. Most of the time we are right. So synthetic biology has been compared with engineering very often, very often. I think that this comparison is a bit misleading for a number of reasons that I'll try to illustrate. But the thing is that to what extent is that true? We'll discuss it. Let me go for one by one with different kinds of systems, okay? The first I'm not going to spend much time. The first I mean protocells. But I do want to mention a very important aspect of what you just mentioned about mathematics. Why mathematics in trying to explain living things? And I think that it makes sense also in the context of the previous talk. In the previous talk, we have been listening to how you have to approach the molecular dynamics of systems to understand experimental observations. What I'm showing you here, you know who is this guy? Von Neumann. Von Neumann was one of probably the last Renaissance mathematician of the 20th century. He worked almost on every area. And at the end of his life, he died of cancer quite sooner than should be interesting for science. He was thinking in several problems related with biology, from how to make a computer that operates somehow like a brain, or what kind of machine would be able to self-replicate itself. He was a mathematician. He was ahead of molecular biology, so no one knew about DNA, polymerases, or anything. But he ended up with a very interesting scheme. He ended up with a scheme that's saying that a machine that is able to self-replicate should contain instructions, should contain a system that he called a duplicator, then a controller and a constructor, which essentially not only replicate the machine following the instructions, but they too replicate the instructions. The whole logic of that is something that if you show to a molecular biologist or a biologist, it's easy to identify element by element what you're talking about. The instructions are DNA, the duplicator is the polymerase and other molecules that help in the process. So it is interesting as a intellectual conclusion that this suggests that the logic of self-replication might be unique. There's only maybe one only way of doing that. Concerning building protocells, this is a story that has a lot of characters involved. I will say that there are two basic approaches to building an artificial cell. One is the top-down. Doesn't work. Anyway, top-down, you start from a living cell. You try to reduce the genome. The other one is the bottom up. You try to, from chemistry, you try to go up and find something that is able to self-replicate. None of the two strategies has succeeded, I will say. For sure, I've been working several years on the bottom up. We learned a lot of stuff in the process, but we didn't succeed. There are a number of different strategies that have been used, most of them on paper or in simulation. And one interesting thing of working on that has been that we have been discovering in the process that there's a lot of complexities that we have to deal with, and we still don't know how to do it. I can mention that, among other things, it's possible to show that a very, very basic system that you can simulate using a basic molecular dynamics can undergo one of these cycles, but this is still a toy model. And if you go seriously into the chemical rules, some of the problems I've mentioned before, this is probably not a realistic one. Just to mention something that I think is quite interesting about complex cells. Complex cells means that through evolution, at some point you have cells that have a nucleus, that have internal structure, because everything seems to indicate there was symbiosis. It was the joining of different cells, which was a quite peculiar event. Lim Margulis was one of the persons who suggested that idea. The guy there is Luis Serrano, who is in my center in Barcelona. He leads the Center for Regulatory Genomics, and they work on trying to figure out what's the logic of the minimal living cells that you can study today. In particular, they use Mycoplasma pneumoniae. And besides of finding out that even those simple organisms are much more complicated than they expected, they try to do something that is kind of one of the first major synthetic transitions that I was talking about. Whether or not you can manipulate one of these cells, put them inside another cell, okay, and that the machinery of that cell keeps working and doing a function that you want. So that's something that has to be done still, but is probably going to happen in the next years. Another thing is, can we engineer existing cells and tissues? And that brings us into the domain of synthetic biology. Essentially here is synthetic biology is the granddaughter of genetic engineering. So we have the idea that we don't want just to change a single gene or put a gene inside the cell, but put whole circuits and try to change, in a way, the logic of real cells so that they do things that they should not be doing normally. The origins of synthetic biology, this is a story of 15 years ago, starts with people building switches. So you want a switch. Why? Because many things that happen inside cells have to do with switches. Your cells in the brain, your cells in the lungs are different. Why? Because different genes are on and off, okay? And that defines the phenotype, among other things. So you can build a switch, you can build something that can be in different states, and you might control that. Switches, then you can build internal clocks. There's been a lot of things that have been done as a proof of principle. For example, designing a bacterium that recognizes specifically one type of cancer cell, and when that happens, they can kill the cancer cell. That sounds great, and it's great, but still far away from going into the ending. Okay. What about artificial cells? Because it was a few years ago, there was a lot of hype about artificial cell being created in the lab. You probably remember that. Of course, this is the journalist who mix everything all together. Is there any journalist here? But the thing is that what Benter, who is a controversial guy, you know that, Benter and his colleagues did was to take a genome that is existing from a cell. They actually rebuilt chemically using computers the same genome, which means that you don't understand anything. You just rebuild the sequence. So they build the whole chromosome, changing things that they wanted so that you can actually take out things that you don't want. For example, genes involved with toxins. They put their names, and then they put inside another cell from another species that had been previously emptied from genetic material. So you reset, it works, okay? is kind of a genome transplant. So it's kind of an exhibition, a tour de force. What is true is that this is an interesting example of what is the meaning qualitatively of synthetic biology. One thing that Benter said, and it's correct, is that for the first time we have broken something that has been happening in the biosphere for millions of years. You know, every cell was coming from another cell. That's not true anymore. From my side, I like a little story of Benter when he was asked by a journalist, people says that you guys are playing God. And he said, who says we are playing? Well, the potential for that is great. And my group is working in a couple of biomedical applications. Here is some hype, but the potential is real. So one thing that we expect to make true in the next years is that in diseases where it's not just a single element that has failed, but a whole decision-making system, for example, in diabetes type 1, which affects children mainly, your immune system destroys your beta cells, so you cannot produce insulin, and it doesn't make any sense to put more beta cells in the pancreas. So an alternative that we are trying to explore is to rebuild the circuitry using different cells from the pancreas, and that seems to be working so far. Another thing that is quite fascinating is the discovery that you can call yourself a species, but of course that's a bit misleading. We have about 25,000 genes, but inside us there's a microbiome. It's a huge amount of bacteria. We had the idea that we have bacteria, but so what? But the thing is that we have a lot of different species that is something that is transmitted generation by generation, is conserved, is preserved. And the microbiome implies a whole ecosystem which goes up about 3 million genes. So you can say that you are a species, but you are not. Okay? And the microbiome opens the door to many, many things. It's changing a little bit our view of disease. It's clearly that microbiome communicates with our cells. So if we are able to engineer the microbiome, and that's what we are doing now, we can communicate with our bodies in ways that are quite simple to engineer in principle. And there's a lot of expectation of that, and I use that example in the end to talk about something else that you will see is quite interesting. Just to mention that one of the challenges of synthetic biology is precisely what have to do to go into engineering ecosystems. You know? The microbiome is not a single species, it's a more complex thing. That has advantages and other problems. So let's go again into the computer picture. Right? To what extent engineering is a good way of approaching synthetic biology and trying to imitate engineering to going into biology. This is another quote from George Johnson, where essentially he says that the cell is kind of a microchip with lots of components, and the question is how entangled is this? So can we engineer life? Can bioengineers avoid tinkering, meaning that evolution has been tinkering a lot? We are the outcome of a blind process. Evolution, as a difference from an engineer can, in principle, do whatever it wants, in principle. In principle, you mean, in parentheses, maybe it's impossible. But in principle, you can do that. Nature cannot. You have to reuse what is there, okay? So you have to tinker. And some things can be avoided, some others cannot. And that's one of the metaphors that appears to be more and more common in synthetic biology, circuits. In the end, inside cells, there seems to be circuits, signaling circuits, genetic circuits. So maybe the metaphor of electronics is good, okay? Well, let me show you why it's not. And that metaphor has been used repeatedly. And just incidentally say that, for example, we know from electronics, from logic gates, that there are some particular pieces that you can use to build anything, for NOR gates, NAND gates. And in this paper, which is interesting anyways, at some point, the author said that you build this gate, and by connecting such NOR gates together, any computer circuit can be built. They forget to say, in principle, because it's not the case. This is a paper that's very interesting in Nature about the problems of synthetic biology. Let me show you what's the problem. Imagine you want to build a logic gate. The details are not important. You have two inputs, for example. You want an output that depends on what you have, 0, 0, 0, 1, 1, 0, or 1, 1. Okay? So you want to design it. For example, an AND gate could be a table that is there. You have alpha and beta are input signals that can come from the medium, can come from other cells. And you have inside a response, which is usually a green fluorescent protein. It's one of the standard things that we use. You see there are some test tubes with fluorescent proteins. And they are using more and more, and they are actually changing our way of imagining biology. Right? There's also one contemporary artist who has a rabbit, which is fluorescent, and he builds a whole discourse from that. I don't see the point, really. But anyway, I guess that's contemporary art. The thing is that you can easily build this gate, okay, just, for example, stimulating one gene, another gene, making the proteins go together and, for example, inhibiting the fluorescent protein gene. And that implements an AND gate. Fine. You will say, OK, you extrapolate. And that's all. The problem is that you cannot extrapolate. That's not so easy. For example, imagine you want to build a circuit that is not really large, very common. It's called a multiplexer. So essentially, it's a band selector system with 3 inputs. On paper, you can go and you can draw lines. Say, OK, imagine that I build this gene, I connect these ones, and these ones, and these ones, and I get that. Okay? What is the problem? The problem is that there's a fundamental difference. The problem is that this is not going to work. I can tell you now that I've been also doing experiments. Every cable here, cables means like connections that I've been drawing like lines. Every cable has an identity. It's not like a copper cable, which is identical for everyone. And every new cable means that it's very likely that this cable is not connected only with the gene you want, but somewhere else. That's what we call crosstalk. That's the demon devil word. Crosstalk is what fucks up everything. So how do you go beyond that? One good thing of being a member of the Santa Fe Institute is that for many years I've been having the privilege of talking to people with very different views of almost everything. And one thing that has been developed at the Institute also is a lot of thinking about computation. Computation in nature, computation as a result of evolution, non-standard computation. And there are a lot of metaphors in nature that illustrate the idea. I know that no one is listening to me now. Illustrate the idea that computations are not just what an electronic circuit does. An ant colony performs computations. It's actually a good example of emerging computation. Single individuals have no idea of what the colony is doing. And you cannot understand what the colony does looking at single individuals. But collectively, they do computations in a parallel way under noisy conditions using different strategies. Sorry, I have to. So we tried to approach this problem in a different perspective by breaking rules. And I think it was an interesting exercise. This is the team that succeeded in finding out a different alternative that says what we call the wiring problem. More wires, more problems. So try to avoid the wires. The team is composed by biologists, molecular biologists, and physicists. And it was interesting as an experience. At the beginning of the project, we went in the office of Francesc Posas, who is there in the center on my side, and with diagrams saying, you have to build that. And he was always saying, but do you think it's going to work? Seriously? And then from our side, we're always asking, are the experiments never going to finish or what? I mean, it will have been a year, in another year. After the project finished, the language totally transformed. So every time we went with a piece of paper, Francesc said, it's going to work, for sure. And when people asked us about how's your project going, he said, well, you know, it takes a while, maybe 1 year, 2 years. The thing is, imagine that you break rules, okay? So instead of building things, engineering everything inside one cell, do it in different cells. That's not new, okay? The multicellular circuit is not new. People has built engineering one type of cell, another type of cell, and they communicate them, and they do something. But imagine you do the following. Imagine that you can break the circuit, meaning that some cells cannot be connected with anything. So there are parts of the circuit that are completely disconnected. Of course, nothing to do with standard electronic engineering. And also that the output, so the cell that has to blip and be fluorescent, can be in one of the cell types, but maybe in another, maybe in all of them. So the output is distributed. Okay? Again, doesn't make any sense in standard electronics. But it works. So the essential idea is that you have the circuit, you have your output, but instead of doing that, you break the circuit. Cells don't need to be communicating at all. Okay? And you can do it in many different ways. And for example, the multiplexer that I just showed you before can be deconstructed in this way. You put two cells, both can have the output, two receive an input, two inputs here, and you can show that this is a multiplexer. And the engineering you have to do inside is completely minimal, completely easily achievable. You can actually extrapolate from that. After 2 years, the combinatorics was on our side, and you can build all kinds of circuits. And even circuits that should be very, very complicated, using any standard minimized circuit, reduced to a very, very simple thing from our method. So what's very interesting from one side? Maybe the engineering metaphors that we consider the standard have to be abandoned. You can actually keep going if you use space also, microfluidics. You can show that you can go into the minimal possible wiring and scale up the engineering and build very complex circuits with not much problems. And in the last phase, we are also working on building cells that can do associative learning. Now, a little bit on the lines of Pavlov experiments, where you train or a given animal, like a dog, for example, learns how to associate different stimulus. So, beyond cells, how did multicellular life emerge? That's a question that has been around for a long time. Big advances have been done in the last 5 years in terms of the phylogenetics, understanding what the origins of the first parts of multicellular life. Multicellularity is also a great example of a major invention because it has been invented many times. It doesn't appear once, it appears many times. And can we create tissues and organs from scratch? Do you know who this guy is? It's in a movie. Not the name. What's the movie? Blade Runner. Who said that? Right. I asked that to my students because my students, for some reason, are not only ignorant in some mathematics that they should know, but they are also ignorant in movies. So we try to correct that. So actually, in the exams, there are questions that address movies, so they have to watch the movies. No escape. Anyway, this guy in the movie is the guy who built the eyes. In Blade Runner, I hope that everyone has seen Blade Runner, okay? If not, you have to. He's the guy who built the eyes for the replicants, for the android-like, how to say that, machines. I don't know machines. At the time that was made, thinking in terms of building eyes was kind of a science fiction thing. The funny thing is that it's not going to be fiction anymore. In terms of trying to build synthetic systems that involve multicellularity, there are a number of approximations that we can use, from computational to real lab experiments. Okay? And my group is involved in a project that, after 4 years, is starting to give finally results, which essentially is the idea of you take single-cell species, E. coli, for example, that they usually do not behave like a multicellular system. You engineer connections, so they have to connect and talk to each other, and see how far can you go. Can you go into a tissue? Can you go into a multicellular system? One thing that we wanted to build is Turing patterns. Alan Turing, which you all should know, one of the big intellectual influences of the 20th century, not only built the theory of computation, but also at the end of his life also, and many people say actually that, that Turing killed himself for very sad reasons. Many people say that if von Neumann and Turing hadn't died so soon, probably science would be very different, because they were actually at the forefront of connecting computation, biology, and things that we are doing right now. And Turing formulated an idea, which is the following. Imagine you have a population of cells, or even a single cell, which is completely homogeneous. How from that can you create order? Can you create structures? Can you create different cell types, organize them in space? Where does this order come from? And Turing ended up in a very original, very interesting theory, which essentially says that if you have two molecules that connect to each other, that can interact and diffuse in space under some quite general conditions, you can prove mathematically that they will create order from disorder. Okay, so you just have an initial noisy state, everything is more or less the same, and this is amplified and you generate structures. This has been proven in physics and in chemistry. So you can show that many things that you can get in the lab that follow the Turing mechanism, this amplification of perturbations, of fluctuations, is there. In biology, there's a lot of indirect evidence. So there's a lot of scenarios where you see, for example, the code patterns of animals or fishes that they have to be Turing. Okay, you see that, but they have to be doesn't mean that they are. Okay, I think in the previous talk we have heard that. I mean, you have a conjecture, but a conjecture is not enough. So people criticize that because they say, okay, what molecule is molecule 1? What molecule is molecule 2? Can you show me that this is the case? So one thing that we want to do in our lab is precisely to create synthetic Turing patterns. So use the machinery that is there, not necessarily the natural one. Maybe we can mix things from everywhere and see if that's the case. And after 4 years, we succeeded. My colleagues tell me, don't show anything to anyone. But anyway, if I'm quick, it was the first result. These are structures that are 1,000 times larger than single E. coli's. And that was just the first result. So it is possible, and it's possible to do quite cool things. Another approach, of course, is computer simulation. And one thing that we believe is very important, and this is our experience by working with models of cells that can evolve addition, cooperation, can evolve their little metabolism, etc., putting them in a physical space under the constraint that they have to eat and find out where is the energy, it immediately leads to very, very interesting results. For example, in this experiment here, what happened is that the system starts with the same kind of cell at the bottom. You receive energy that drops from the top. And under a process that is essentially trying to maximize the surface to gather energy, that leads to a leap. A leap is kind of an innovation where the cells actually discover that the energy comes from the top. So they go to the top, and then start to do other things. Like, for example, develop aggregates that cooperate. And you keep going. I mean, it's possible to build systems with genetic networks, and that's ongoing stuff. But again, remember the automaton I was showing you at the beginning. It's an automaton that has a body. Having a body, having the physics, it turns to be quite important. Two things that I think are quite remarkable, and again, tells you about how do we have to rethink some ideas. You have heard about stem cells, and you probably know that now, today, you can take stem cells from your skin, reprogram them, which essentially means that you put inside the cell genes that are inactivated or even can be just not present. You put them, and the cell becomes a stem cell, right? And under the right conditions, you can make the cell differentiate into whatever, okay, whatever you want. That was a surprising result, because I think that 10 years ago, everyone would have said a differentiated cell is an irreversible thing. So this is the result of a cascade of changes that is essentially irreversible. So that tells us that something was quite much missing about our models of genetic networks. The other thing I want to mention is that is regeneration. Why we don't regenerate a finger, for example? Well, there's a good reason for that. If a salamander or other animals can do it, we have the evolutionary trade-off is that it's much better if we try to seal bounds, okay, to be sure that we don't have blood escaping from our body, and instead regeneration has a drawback. I mean, if you have mechanism of regeneration as potentially active, the potential also that you develop diseases like cancer is much higher. So what happened? Does it mean that we cannot regenerate? Because one thing that evolution always leaves is a mark. Almost all the evolutionary history is there. And in the same way that some people were able to induce the creation of teeth in hens, in chicken, that chickens have no teeth, you know that. So they could regenerate that, and the teeth are quite interesting. Are not the teeth of a mouse, something else, something that probably has to do with predates the origin of that particular group. And the same happens with regeneration. One thing that has been found out, and this is so recent that I think that there are almost no papers on that, is that there's this particular story of this guy who was playing with a little plane, and the helix cut his finger, and he couldn't find it. So if you find it, you go to the hospital, and you're lucky. They just put it again. He couldn't find it, but the chance was in his favor. In his circle of people, there was people who were doing experiments using so-called extracellular matrix. The extracellular matrix is part of our tissues, and it's very important to send signals. Okay, and they have been using that experimentally with a number of animals that had been, seriously harmed, and they tried with him. So they essentially put this extracellular matrix, which came from pigs. There's no reason for that. It's just, And what happened was that you put this extracellular matrix, and you send a signal to the stem cells that usually do not respond to anything like that, and everything regenerated. So which means that the whole finger with a nail and the fingerprint. And so that tells us also that, that we might take advantage of things that are there. And concerning the guy from Blade Runner, what about the eyes? Well, one thing that has been found out is that self-organization, the whole machinery that is ready there to create structures, to create organs, okay, you can take advantage of that. And if you put the right stem cells in the right medium, you can see that from this homogeneous initial condition, stem cells reorganize, adhere to each other, start to reproduce, and you end up with an eye. It's an eye which is, in many ways, is a prototype, but is complete. And the same happened. You might have heard about that. I think it was last summer. If you do that in the context of brain cortex, so you can actually do the same experiment and observe the formation of this kind of micro brains, right? Which, if you slice, you can see this kind of cortical structure. Some journalists, at least I remember in Spain, I remember one paper that said, "Where is the consciousness?" Well, journalist, is there any journalist again? That's not interesting for that, but it's interesting because it's a unique window to see things that happen within the process of creation of the brain. As the fetus is growing, the brain forms, and we can have a great window into disease we didn't have before. And in terms of an engineering and design, means that if you want to think in engineering these kind of things, these tissues, you have to rethink the way you approach that, because self-organization is there. So you might work on that, but take into account that there's something on your side, which is the self-organization that is already present. In another aspect, you can build, you start to be able to build organs. Many interesting things have been already applied in medicine, for example, building a trachea. And another thing that I find fascinating, and again, is kind of a breaking one thing that happens always in biology, that is organs and parts of organs are the outcome of morphogenesis, of embryogenesis. But you can actually print an organ. So you can actually use a 3D printer with cells, and that has been done already, and build organs, which again it's an interesting possibility. We make a big jump now, okay? This is a big question. Some of them have been raised at some point here. This is, for me, a very, very interesting question. Unfortunately, there's a lot of crap that is being said all the time. Can we create intelligent consciousness? What the hell is consciousness? Okay, that's the philosophically well-stated question. More important, I think, are these the right questions right now? And I will go to that. Can we create a synthetic collective intelligence? Well, again, Turing, right? He was the one of the first who formulated that, tried to put that into a scientific context. So how do I know that a system is intelligent? How do I know a machine is intelligent? Okay, and he formulated this idea that you probably know, this Turing test. It was called after him. If I'm typing, I'm connecting with a machine, or I'm connecting with someone. By typing in a computer, I type questions. For example, do you believe in God? And then I receive a message, okay, on the screen. I can maintain a conversation. The question is, at some point, can I know if this is a machine or is someone else in another room who is actually answering? Okay, if it's a machine and I think it is a person, then it has passed the Turing test. Okay. A funny story that was told to me to Krakauer, David Krakauer from the Santa Fe Institute, is that, of course, these machines have to be, if there's a machine, have to be ready to a typical question. For example, you can ask the machine, do you believe in God? It seems kind of a trap. But they are prepared to do that. So he told me that he was at MIT, that they put two of these machines communicating. And it was really fun, because they started to ask things. And at some point, one of the machines asked, do you believe in God? And, and the conversation afterwards is really amazing. I have to say, much more interesting than many conversations I hear usually. So is the right question right now asking about, can we build a machine that is intelligent? You know that Isaac Asimov was very much a pioneer in thinking about what would be the implications of having a machine that is, to a level or another, intelligent. Okay? And there's movies and everything. But in the process of trying to figure out what can be built, one thing that is fascinating, for example, is what evolutionary robotics can deliver. And that's one of my preferred examples, for example. This was a work done by Dario Floriano and colleagues, Sarah Mitri and Lauren Keller, where actually they use robots that can evolve their neural networks. So I'm telling the story very quickly. You have a given area where you have sources of charge. So if you go there, you get charge. So for the robots, I guess it's fantastic. And places where you receive a discharge. By discharge, you mean electric discharge. So you have to move around. Initially, no one knows anything. You have also lights, red and blue, like Barça. And they move around, and they learn where is the source of energy, charge, and discharge. Okay? Two things evolve, among other things. Okay? One is cooperation. So the robots eventually send a blue signal if they have found a source of charge. So come guys, here you can charge yourself. And a red signal if they find out a source of discharge. So they prevent others to fall there. But what happens if you're a robot, okay, and you go to the source of charge, and it's full of people around, and you cannot be charged yourself, right? That's not good. So an outcome of the process that evolves is what in game theory is called cheating, for example. But in this context can be said is lying. So they send the wrong signals to the other robots. So come here. Yes, it's fantastic. And they go and they get discharged. Okay, or there's nothing to see here. Yeah, circulate and there's the source of charge. Quite interesting. Another thing that is quite interesting is this guy at the left is Luke Stills, who is in my institute. Some of you might know him. He was one of the great pioneers in evolving robots, in particular in the context of language. You will hear about language tomorrow, but so I don't want to give much details. But what very, very interesting result from the research that they did, that they keep doing, which is having robots that perceive the environment, that the embodied robots, okay, and they communicate to each other, and they can name things and then agree about the name of the things. Is that one of the outcomes of that is that they evolve a protogrammar. So it's not just developing an inventory of words and referring to the same things in the same way, but also evolving the first part of what we'll say language. And of course, there's a big number of questions that emerge around that. And one of the ones that I find most fascinating is there's a robot, it's not one of these, that's called the iCup, that's been used for many, many experiments about how you develop things like a child. There's people who start to believe that maybe being a child, learning like a child is important to develop language. two recommendations. You see that I like movies. Two recommendations that I think that you'll find very interesting. Robot and Frank is a movie about a guy. I'm not going to tell you the end, okay? About a guy who has started to have Alzheimer and has a very, very interesting past, so to speak. And the children of this guy decide to give him a robot so that take care of him. So it's a really interesting story about the potential. The robot is not intelligent, but there's a relationship and there's something that makes you think about what's in the near future, about what's going to happen when we have robots that can learn about us, that can change, that can, it's not like a cat or a dog, with all my respects to both, okay? You can talk with the robot, and if the robot is not intelligent, what is going to happen with that, okay? And here is a kind of the other part of the story. So what happens if you have an expert system that has kind of intuition, so to speak, and that you can also communicate in complex ways? Is this another organism? Is an expansion of you? And what's going to happen in general, okay? And that's why I was saying that what are the right questions, okay? Wittgenstein, in this particular quote, says something that I find very, very interesting. Could the machine think? That's what we are saying now. Could the machine think? Could it experience pain? And that's a quite deep question. How do we model pain? What, for us, pain is extremely important. It's another big invention of evolution. Without pain, we will be unable to survive, I'll tell you. But what will happen if we develop a robot that experiences pain, for example, and that, or can experience fear? Fear and pain are things that you will not say that this is intelligence. But would you be confident or comfortable in putting the plug out of that? That's another question. So we might not build synthetic intelligence right now, but we can do something in the steps of another kind of intelligence, which is sociality and collective intelligence in insects, which is another big area, which I know people is working here. In robotics has been the inspiration for a lot of collective robotics. So how do you manage to do robots to imitating the rules used by ants, for example, to solve collectively problems? And they feel advanced a lot. That's actually an old picture. It's a robot which has sensors, can be programmed, has an internal clock, which of course was built in Switzerland. And you can use these things to do enormously interesting experiments. Some of my colleagues in Brussels and France use that to manipulate, for example, the behavior of cockroaches. Cockroaches are these disgusting insects that are not social insects, so they are kind of gregarious if you want. But you can use robots to manipulate their behavior so that you force them to behave collectively and solve collectively problems that are usually solved by ants. So it tells you something about the preconditions, that there's a lot of things that might be there in the biology that you can use actually to make a transition. So we found out some time ago that actually some of the mathematics we were using to model genetic networks were very close to the mathematics that we've been using years ago to model how ant colonies solve some problems. For example, finding out the shortest path between two possibilities to a given food source. So one thing we decided, and that's the other big thing we are doing in my WETLab, is to try to create synthetic collective intelligence. So is it possible to manipulate bacteria so that they behave like ants in such a way that bacteria collectively can solve a problem, like a short path problem? We got a lot of inspiration from the collective behavior of animals in nature. There's a lot of good models that capture a lot of the global behavior that we observe. And we are, I think, that 80% on the end of that. And we believe that, yes, it's possible to manipulate bacteria so that they behave like kind of little robots. Just to finish, there's another issue that has emerged. It's my last point in the list. Will we survive? It has to do with something that has been raised here, the problem of climate change and our current position in the biosphere, right? Synthetic biology offers the potential for many things, okay? Some of them are fascinating. Some of them are going to be important in biomedical applications. Some of them are under discussion. For example, would it be possible to use synthetically designed organisms to solve environmental problems? So you have a place where you have a toxicity by some given waste, and you have real problems to isolate that and extract that. Could we design organisms that can do that? Of course, this raises a number of problems. For example, can you just free these organisms to nature? And that connects with, that's the final century. That connects with two things. One is the ongoing view that our Earth is approaching some transition. And that means that we know in complexity that almost nothing in a complex system reacts all the time linearly. There is typical behavior to have shifts so that you react in a nonlinear way. Many things that are going on in our planet already point in that direction. We heard this stockfish collapse. That's one thing that has been happening repeatedly, and unfortunately, that doesn't percolate into the mind of politicians. There's the prediction that if climate keeps going into the wrong direction, it's going to be a turning point. Okay? I agree with Professor Rees was saying that we need still time to know if the predictions are going to be the best ones or the worst ones. Okay? I have a worry about the averages because the fluctuations can kill us before the average is okay. And so the question that I want to raise, and that's a conjecture I've been thinking over the last years. It's about an alternative. It's been discussed about geoengineering, and it was pointed out also the problem that geoengineering has a lot of side outs that we might not want to experience. There's a very good review paper in the Royal Society by Stephen Schneider, who was really a pioneer in many of these areas. And the proposal that I'm making is based on what we know today in synthetic biology, based on the fact that we might not be incapable of designing things that might be the right things that we want to design, whether we could do something that scales up with the biosphere. So geoengineering has one problem, clearly, is that I have to operate at a local scale. Otherwise, the cost will be unthinkable. How do you reach the scale of the biosphere? Well, a living thing can do it. Okay, a thing that once freed in nature can expand and reach a large scale. Doesn't need to be the global scale, but large scale. So the idea will be to build a synthetic organism or organisms that can reach global dispersal. There's a number of good candidates. You have to design them to limit evolvability, because if you evolve things, you are free from evolving. From Jurassic Park, that sentence that life finds its way. Who knows what's going to happen? My conjecture is not much. Probably what's going to happen is a failure. Limit evolvability. There are numbers of doing that. Integrate it within the biosphere. That means that we could integrate that in the long term. So organisms that could live inside sponges, for example, which stabilize the evolvability, or maybe things that we are working now, for example, on systems that build biomaterials, and once they have made their function, they die. That's another possibility. And I put last, but maybe should be the first, think about it before doing it. So why or why not doing that, I don't know. But I think it's been interesting that at the beginning of this conference, the Manhattan Project has came about, because there was a big project that joined the minds of many people to try to solve a really big problem. Unfortunately, it wasn't very creative in favor of people. But my conjecture is that the day will come, now I look like a TV preacher, the day will come that that kind of scenario has to be considered. And I think that is a good time to start. Let me finish with two sentences. Since God and everything has been mentioned several times, I want to make my point, okay? As an atheist, okay, I think that this is a good description of what I think. God is the silence of the universe, and man is the cry that gives meaning to that silence, which I think is a sentence which tells us about how small we are in the end, about how great also. And about complexity, I think that this is a very nice sentence by John Wheeler, one of the giants of the physics of the 20th century, who says something that is so clever. You know, we live in an island surrounded by a sea of ignorance, especially politicians. As our island of knowledge grows, so does the shore of our ignorance. I think that working in complexity theory means that you have to really risk yourself going to the shore to see to the beach what is there. Maybe a shark, maybe you'll not find water, but this is the most funny part of the story. So leave the jungle, go to the beach, and you will enjoy it. Thank you.
Thank you very much for. All right, thank you very much for this amazing talk. Normally I come to talks to have answers, and today you gave us a lot of questions. I think we are in business for many, many years, and especially for the Complexity Institute. Any question from the audience? Peter, please.
Wonderful talk. Many thanks. So there is a danger that with this synthetic biology, we start to think that we understand things which we actually do not understand. So let me give you an example that comes back to one of the things that you mentioned, and I would like to have your opinion on this. So the work of Turing on these striped patterns, which quite neatly explain how these striping patterns come about, and that were compared with the stripes of zebras and all the things like that, was completely wrong. Because in the end, what was shown by Nusslein and Volhard was that it's actually the homeobox genes that drive that whole striping pattern. Now, so what you quite often see in complexity, and also to some extent in this kind of research that you talked about, is that we generate something that looks like something that we're trying to understand, and then we say, okay, it's the same. But like you can generate a zebra stripe pattern by a Turing pattern, but that's not the mechanism that's underlying the actual Turing stripes, the actual zebra stripes. So, can you comment on this a bit and help us to, because the work you do is absolutely amazing. So what I want to understand is, can we really distinguish these things? And are we not in the risk that we run into a trap that we think that we understand something which we actually absolutely do not understand?
Sure, yeah. Well, first pointing that Nusslein and Volhard work, it's a great pioneering stuff, has a lot of years. You remind me for a moment, one of my first supervisors. I started 2 PhDs that I left. And one of them was, my supervisor was all the time saying, you see, Drosophila, this Turing stuff is crap. It's never happened to be found. Have in mind that Drosophila, which is what Nusslein and Volhard worked a lot, has this segmentation pattern that we know is not Turing. It's kind of a hierarchical mechanism that actually is, if you study the problem from the general perspective, there are 2 main ways of generating patterns, which is hierarchical, so you produce a given collection of things in a sequential way, or self-organized, which means that you take advantage of amplification processes. We observe both of them. And in some cases, you fairly well identify which are the genes involved in the process. In the systems where you see that cells, in a way, use this information to amplify fluctuations, you see that indirectly. And there's also the problem that often you have intermediate steps that fast completely the simple mechanism. There are not such a thing as a simple Turing mechanism, for sure. And then all the things related to growth of cells, growth of tissues. So it's a number of things that you have to add to the whole picture. That said, I think that the evidence in favor of mechanisms that rely on amplification, it's very clear. So this self-organization is there, no matter if you know exactly what are all the pieces. And then both coexist. Okay, I will say that right now, if you look at the whole picture we have, I will say that the self-organized part dominates over the hierarchical part. So to say the Turing part would be unfair because Turing is a specified thing. But this self-organization, that's for sure. And I think that now in the next year, we'll see more and more evidence about that because people are identifying the players. The potential for doing that in synthetic biology, I think that besides of offering the opportunity of going into the minimal mechanism with very well specified players, is that from that you can go into many other directions. And we're just finishing that, but it opens the door for many other things.
So, yeah, I guess I'm just a bit worried about that we end up with a science like that consists of only metaphors, where we say this is analog to that because it looks the same.
Yeah.
Actually, a friend of mine, Dan Frankel, used to say that. He said people in complexity, they say this is complex and that's complex, so it should be the same. And
yeah.
and well, that doesn't really help us.
That's a caricature of complexity.
Yeah. So, but you gave wonderful examples where you can actually test these things. So could you comment a little bit more on this? Because I think this is really in the crux of this whole conference.
You mean for real biological systems? Yeah, in my own building, there's this group led by James Sharp that they work on the morphogenesis of the limb. And they have published last year in Science a paper where it actually show that as far as they can prove from experiments and perturbation experiments, and that's the interesting thing, because I completely agree with you. If you have a pattern and you have a model and they match, that might not mean anything. So you need always to have several orthogonal things that you test, because if not, for sure there's an alternative. And what they do is precisely that. They do experiments where they perturb the process of generation, and they can find out that the expectation from a model based on amplification of fluctuations is completely consistent.
Thank you.
At the beginning of your talk, you gave these lists of 10 key steps in evolution. Would you like to say a word about your views on contingency versus convergence? Yeah. How much of what happened is random, or would things inevitably have ended up rather like they are in our biosphere? Yeah.
That's one thing that also I think is an important threat in both in evolutionary theory and if we want to make a kind of a theory of artificial evolution. In biology, we know that, and there's been a lot of discussion about. Steve Gould was a very strong advocate on if you go back in time and you put the game back, only God can do it, of course, the tape of evolution from this Cambrian explosion, or before than that, you will observe essentially a completely different biosphere. And instead, you observe something that is interesting. I think there are two ways of approaching that. One is that this apparently widespread role of convergence. You go into a desert in Australia and look at the seeds of some plants, and they have this very peculiar structure with these spines and this structure. And you go to another continent, and you find exactly the same solution. And that happens really repeatedly. And moreover, in the context of major transitions, multicellularity and other things have been invented several times. Okay? The eye, for example, for reasons that for me are very clear, the octopus has an eye which is great as ours, okay, except that his design is the right. Okay, so you receive the light, they go into the receptors and then to the nerves and the nerve. So we have the retina is kind of how you say, reversed. Okay, so you first the nerves and then okay. So you still want to believe in God, believe it, but it was like he was sleeping that day. It was the 7th day, I guess. Was tired. So convergence seems to be widespread. I do think that it's important to find out whether or not we can make a theory of that. So why is some solutions maybe unique? So repeated that if you go to another planet or we go back in time and repeat the story of evolution, my conjecture is the biosphere will be different, but familiar, clearly familiar. And in an interesting experiment that was done some years ago, with some limitations, and we are trying to go back to that with our artificial life experiments with physics, is those experiments by Tom Ray, And actually, the first guy who did that was Niels Barricelli from Neumann. Using computers, you simulate how population evolves. And the outcome of all the limitations is spectacular, because they started with a collection of programs, identical programs, with just replicate and compete in the memory of the computer, but they can mutate. And what Ray found, very briefly explaining, is that you have selection for shorter programs, because you're smaller, you replicate faster. Then there are programs that cannot replicate by themselves, but use the other programs, so the parasites. Then hyperparasites, and then something came out, which was kind of sex. So much more boring than the human one. So essentially, it's programs that exchange part of the code, so that they escape from parasites, which actually recognize specific sequences. And eventually, cooperation. The groups of programs that one by one were not very efficient, but cooperating, they did. So this is a complete different scenario than the real biology, but tells us something very, very, I think, fundamental, which is at the level of the essential logic of what to expect to emerge in a biosphere. Parasites, sex, cooperation might be inevitable outcomes. We are far from having a theory of that, but I think that maybe we are in the good track. And I think synthetic biology can help, because in a way it's not theory, it's experiment, but it's an experiment that you predefine by hand. And in a way, it's a simulation too, but it's a simulation with living material. So it occupies a very interesting position. So I think that there's plenty of possibilities.
Hi, I have a question. It's I'm kind of curious about the part when you are actually doing the experiment on using the regulation on the transcription and translation mechanism to build a circuit, and you said it doesn't work. However, in the biology world, actually, it is a very important way of forming the biochemical regulatory pathways. So what's the difference between the natural one and the synthetic one? Why do you think it doesn't work?
You're referring to what specifically? What part of the machinery?
Like P53 network. The P53 can actually trigger the translation of like WIP1 and several other, yeah.
Well, you mean if it can be used for synthetic biology?
Yeah, well, why do you think when we are synthetically using this kind of mechanism to try to build a circuit and it doesn't work? However, this kind of circuit is actually persistent, actually work in the natural world.
Yeah. Well, the other thing is that one is long to explain, so maybe we can discuss that later. When you analyze the regulatory machinery, that is heterogeneous, that doesn't mean if it's scale-free or not. If it's heterogeneous, you have these hubs which have lots of connections. Our experience with that is that they play a role like ambiguous words in language, which also define a network. And that provides an explanation, so to speak, of their relevance. I'm compressing a lot of stuff in a sentence, so I'll tell you later. I think I can tell you is that one thing that we tried to do for several years, and it's again something that tells you something about what can be done and what cannot be done, was to build a genetic circuit that showed chaos, chaotic behavior, right? Which was an interesting idea because we knew that we could generate oscillators, and why not chaotic dynamics? So we tried for several years, and the best candidates for that particular target involve P53. For one reason or another, we kind of think we were trying to manipulate. But everything that goes into P53 means that the cell will go into the apoptosis, so kill itself. So that was a good, interesting lesson to learn about the things that we cannot do. Okay. So for the highly connected genes, if that's what you mean in terms of using them to do some things, I will doubt quite much, yeah.
So essentially, it's the complexity of the cell that prohibits you to use it for your own purpose because you don't know what other things you would trigger.
Well, I'm not sure if I understand the question. One thing that I maybe didn't make the point clear is that a lot of the synthetic biology has to be built, the designs have to be built in a way that do not interfere with the rest of the system. which might be very difficult because in terms of the metabolism, you might add something to the metabolic burden. And sometimes this even changes the logic. And on the other hand, if that goes into an organism, but you want to engineer the cells that you want to engineer, you have also to choose the right candidates. I'm not sure if I'm answering the question.
I think essentially that.
I want to follow up on the question by Martin Rees on the uniqueness of evolution. Have you thought about the uniqueness of human history? Could you extend, let's say, dynamics that we have seen in human history? Is there anything like, would there be a regularity there?
So, in other words, to rephrase that, could be possible to make an artificial history that models.
Think about the Neolithic revolution, which seems to be having taken place in several places independently and have had very similar effects everywhere where it took place. Think about the evolution of art, evolution of politics.
Right.
All about that.
Yeah. That's another thing that we are very interested. We have a whole research program there on evolution of technology. A lot of it inspired by Brian Arthur. And of course, here raises several questions about, again, path dependence and convergence. And let me go for technology for a moment, because technology is quite interesting in that respect. There are clearly, and Brian was the pioneer of actually illustrating that. And I like it when I saw him in an interview, and he said that when he went to Santa Fe and started to talk with the physicists, his economics conrads that didn't give a shit of what he was saying, but the physicist was like, of course, yeah, you have symmetry breaking, things like that. And in technology, you see this kind of divergent behavior. You have several solutions, multiple solutions, you choose one, sometimes accidentally, okay, like the clock and everything. But then there's convergence. Convergence is quite spectacular in some respects. When Edison patented his light bulb, there were already 20 patents, which essentially, in terms of was the goal, were the same idea. Of course, Edison was kind of mass production guy, and he was able to find out the right thing. And that's kind of a repeated pattern. I don't know what Brian will think about how repeatable is technology. But this is a question that has been raised in the last years. I think that Santa Fe is an specific interest about that. How predictable is history? Is it possible to build an artificial history as a theory? And that's not new questions, of course, because people have been asking that for a long time, that whether or not history is the result of inevitable tensions that no matter who is the guy there, the tension will resolve into a conflict and the outcome is predictable, or is really path dependent, which is sometimes the way the historical events are described. There's a hero that is responsible for things. And I don't know who was, I think it was Peter Turchin who said that, I don't know of a history of a theory of heroes. And my personal view, but this is an intuition, of course, it's not grounded on a very high knowledge, is that history, to a large extent, is something that follows a dynamical system, and that despite the potential paths that can follow, there's one that is likely to happen.
To comment on that, Ricard, we ran an experiment. This was at Fuji Xerox Palo Alto lab about 6-7 years ago, Wolfgang Pollock and I, where we took not AND gates or NAND gates, and we allowed them to keep combining, and if they formed an interesting circuit, we threw that into the soup for further combination and so on. So in our computer, we had an evolutionary system for more and more and more complicated technologies. The nice thing was that, there were random seeds. We could restart this and see what happened. The interesting thing was, on the one hand, and this is in response to your comment here, on the one hand, we did tend to get the same types of circuits coming out each time. It was like running Steve Gould's tape again and again and again, but they didn't emerge in the same sequence. And because circuit organisms were built out of previous organisms, very often the things that did things were built out of slightly different components each time. So it's as if you went to Australia or you went to Denmark, you found that some things were doing pretty much the same thing, but if you looked into the way they were actually built, the DNA or the modularity of how they were put together, the genetic regulatory network sense, you'd find different networks.
Yes.
So anyway, that's a lengthy answer to your question, but I think those sorts of questions can now be answered in artificial evolutionary systems. And incidentally, thanks for this lecture. Incredible.
Thank you.
We can take two more questions.
Yeah, going back to the question of repeatability and historical contingency, as one of the fathers of the major transitions, I think that I have to say that those transitions that have been operationally unique are genuinely difficult. So it's not only that it's difficult to understand them, but I think that they have been very, very difficult steps for evolution. And there are two that are really standing out. One is the origin of the genetic code. The other one is the origin of the eukaryotic cell. I mean, once you have unicellular protists, the multicellularity is not such a big deal. This is why it happens so many times, right? However, you know, whatever scenario you take for the eukaryotic cell, it's so bloody full of thousands, not dozens, but thousands of sequential idiosyncratic steps, right? So it is true that you cannot say that the eukaryotic cell had to come once you have bacteria. There is absolutely no guarantee that it should come if you repeat the experiment. So this actually generates a prediction. The prediction is that if you find life on other planets, most of them probably would be stuck on something like an RNA world level. A tiny fraction of them would have something like the genetic code. A very tiny fraction of those will have something like the eukaryotic cell. And to finish it, you know, an even tinier fraction would have something like intelligent life. This is what I think.
Just a irresistible, obvious last question. So is it safe to pursue synthetic biology without also thinking about the connectedness of the world, about the last questions that you raised? In other words, evolution is always coevolution, and here we are converting the rainforest into oil palms and the pouring nitrogen, really screwing up one system after the next. So it's a little scary to think about. I mean, synthetic biology is so promising, and yet we are at a tipping point. So do you think, in an analytical sense, do we need to have a dialogue between those two perspectives on the world?
Yeah, I mean, I think it's the time now. Synthetic biology is starting the exponential phase. So it's, so far, everything has been proof of concept, including our work. So if what we built doesn't work anywhere else but in the Petri dish, fine. And to a large extent, anything that's been built, even if it's from wild-type organisms, it's going to fail miserably as soon as you release it. And everything that goes into the biomedical application is going to work inside your body, nowhere else. And the potential is great, and already breakthroughs are happening now. And the thing is that to go into delivering into nature something that has to be integrated, that's a main thing. And I do believe, from my experience in ecological theory, is that there's a lot of things that we have learned about invasions, mutualisms, the way energy flows take place in ecosystems, and what parts of the energy flows in ecological networks are more important. To have intuition about if you want to design something, where and how to put it. I do think that's going to be possible. The big challenge is, on the one hand, being successful if we need that to happen, and on the other hand, that it's not going to evolve in a direction that we don't want. That's the main question. But I do think that it's important to start thinking now, whether or not it's going to happen. Personally, I hope it's not going to happen. But on the other hand, I cannot see how someone is going to ask itself, let's do it.